I wouldn't worry. With continental drift, Africa or South America should come by eventually. A few million years. Lamarck, because he believed that evolution was part of a grand plan, believed that features that were useful in new conditions would just appear. That you'd get what you need when you needed it, because it was part of the plan. As people started to do evolutionary research on small, quick, reproducing organisms, and then they would mess with the conditions, they would often find that features that were useful for the new conditions were there when they changed the conditions. And they went, well, okay, we don't really believe this is part of a plan, and they're getting it, but why are they there? And people went, well, you know, they've probably been there and just weren't obvious, they weren't useful, they kind of came to the forefront when the conditions changed. And other people went, well, yeah, but if they arose 15 generations ago, they'd be gone by now. Is that they don't give an advantage, then they would just have kind of frittered away. Well, Hardy and Weinberg, statisticians that they were, said, nuh-uh, is that an allele will hang around, a new allele that potentially is useful, but isn't useful yet, will hang around as long as there are certain rules that apply to the allele and to the gene pool, the population that the allele is part of. So this has to do, oops. So an allele appears in an individual in the population, right? There's a mutation. It changes the function of a protein a little bit, changes the feature that the protein produces. But under the original conditions, it doesn't help, it doesn't hurt. It doesn't help survival, it doesn't help reproduction. Which means that there's no push to make it go away. So that's the simplest approach. As long as there's no push to make it go away, there's no reason why it wouldn't hang around. And once the conditions change and it becomes useful, now natural selection will make it more likely to get passed on to offspring and produce it at a bigger ratio in the population. Now keep in mind, populations grow. So the allele appears in an individual and gets passed on to some of their offspring. So now the allele exists in larger numbers, but it's kind of at the same ratio within the population. Maybe a little bit, if there's other issues that where the parents or other alleles in that particular genome are useful, maybe it becomes more useful. This is what they call piggybacking. When you give a kid a piggyback ride, sometimes there are features where you look at the feature and you go, well, this is not only not useful, it's actually harmful. But it seems to have stuck around for a really long time and actually has become fairly common in a population. Now, sometimes it's hybrid vigor going on and it's useful in a way you don't know. But there are times where it is closely linked to a very useful allele, and so it just keeps getting passed on as part of the package, and that's considered to be piggybacking. As they find more and more about targeted usefulness for alleles, this doesn't turn out to be as common as they used to think. But it used to be the obvious kind of, well, it must be piggybacking. For the population, you want the odds to work for you. An allele that's rare in a small population can disappear just because, you know, something bad happens to its possessors that have nothing to do with the allele. You know, they get caught in a bad storm, or you know, a disease comes through and just happens to wipe out. Generally, the alleles are in families, so you know, things happen that wipe out families. But if the population is large and the alleles spread amongst multiple families, then the odds of something just chancy making the allele go away are less. Same, you know, it's the same basic concept for when you're doing research. You want lots and lots of subjects because chance will have a bigger impact on a small group. So you need a large size population, a big gene pool. Oops, I left one out. Sorry. It goes in there. Sorry, totally my fault. Once the allele was a product of a mutation. Obviously, you can't change it again. So that allele needs to persist unchanged through multiple generations. But keep in mind, mutations are not that common at a particular locus. A particular allele is unlikely to mutate a bunch of times over the course of just a number of generations. So this was not a really big deal. But basically, once it's in place, it kind of has to persist in its changed form and not change again. Not a really big deal. You get a population. The alleles are probably not evenly distributed through the whole population. So if a splinter group of your population goes off and separates out from the main population, maybe they take the alleles with them. But even if they don't, the ratio of the alleles you have left is now grown. So it does change it isn't an equilibrium anymore. It can become more common if it doesn't go with a splinter group. By the same token, if something changes and a subpopulation that used to be separated from the main population comes back in and merges with it, or at least connects to it, now your gene pool has suddenly increased in size kind of overnight. And the ratios of everything are going to change because the alleles ratios have been changing over here, and your allele ratios haven't been changing to match them, right? The whole idea of mutations are random and they don't appear in the same population. Now, there are a number of concepts that are related to Hardy-Weinberg. There are times where you have a population here, you have a population here, they're separated. We'll talk about isolation later. But they're different. Is that the species are different, even though you look at the ecosystems, and the ecosystems seem to be basically identical. So why are they different? Because once separated out, mutations appear over here that didn't appear over here. Alleles change over here that didn't change over here. And the alleles can be useful. Keep in mind, once an ecosystem, once conditions stabilize, there's still a lot of fine tuning that happens within that ecosystem as far as evolution is concerned. But advantages that appear in this population are not going to match advantages that appear over here, since the advantages are connected to random mutations. And the two populations will drift away from one another in terms of certain select features, like trying to compare leopards to jaguars. They've been separated for a pretty good long time. They are similar animals with similar jobs in similar ecosystems, but they don't look the same. Is that leopards are more yellow, jaguars are more orange, the spot patterns don't work the same. There are some other differences that do have to do with the ecosystems as well, whereas jaguars are in a wetter ecosystem, they're happier with water, they're better swimmers. Leopards are a drier ecosystem, so it hasn't been an exact match. But, is that you see genetic drift in separated populations, probably more common when you're comparing lake organisms that have managed to get into different lakes, but the lakes themselves haven't had a lot of cross connection for a very long time. And you'll find different species in one versus another. Sometimes conditions change very drastically, and most of the population isn't going to survive. Extinction is wiped out. Way more populations have died off than are existing right now, and so a lot of species have gone extinct. Well, what happens if a species almost goes extinct? You have this humongous gene pool spread across a large area, and something happens that reduces your gene pool to just a small population. You took all these alleles that were not evenly distributed. Remember, when we talked about migration, they're not evenly distributed. And you may have reduced your population to a small group in just one spot. Their mixture of alleles is not going to match the distribution of the entire old population. But that's now your population. That's your starting point. Those are your ratios you're going with forward. You've hit a bottleneck where the population was big and diverse, and then it becomes smaller and more limited, but that's what's giving rise to the rest. Happened with people. Is that, I forget how many millions of years ago, there was an expansion of our genus, the Homo genus, across the world, and something happened where they largely went extinct, and the Asian groups apparently went extinct, the European groups went extinct, the Middle Eastern groups went extinct, the North African groups went extinct. Basically, Southeast Central Africa seems to have been where the survivors survived. The bottleneck reduced man to just a single group in Africa. And then they spread out again. What would they look like today if that hadn't happened? What if the groups that had already distributed across the world continued to evolve their own genetic drifts, right? A lot of what makes different human populations different is genetic drift. It isn't, you don't necessarily look at a trait and go, oh, it was an advantage in this environment. Skin color, you can connect to that, but a lot of other stuff that makes human races superficially different. They're superficial genetic drift differences. But you see this. Cheetahs, at a more recent time, seem to have hit a basic bottleneck, as most of the surviving cheetahs are fairly closely related to one another. So it may have reduced the cheetah population to maybe one family. And so now there's a lot of bad recessive that match up when breeding cheetahs becomes a really big issue, because you get what are called lethal genes. Mutant alleles that are recessive produce a non-working protein, but are rare in the general population, but are more common within a related population, a family. So they're more likely to pair up. Now you got no copies that make useful proteins. Tends to kill things. A similar effect that's going to lead into the next part goes back to migration. So a splinter group, a small subgroup of your big gene pool goes off and sets up shop somewhere where they are now separated out. They take their allele mixture with them. That is now their starting gene pool. And everything from there grows out of those ratios, the founder group ratios. And people, you see that in Native Americans, where they don't exactly match the allele ratios of East Asia, which is, their originating population, because a small group went and, probably just a couple of groups with related families within them, so that would have been a different set of ratios. And that was the starting group for North and South America. So type A blood, relatively rare in East Asia, more rare in American indigenous, because there just weren't a lot of A's that went the way. Or maybe, maybe the right number went and one of their, family's boats went under, and that drastically changed the ratio of A alleles in that starting population they built on. So that's going to lead into another aspect of genetics and evolution that we're going to cover in the next video.